Hello everyone, this is the next session of BioTalknology. Welcome. I'm Nupur, a co-founder at Premas, and today we have with ourselves Dr. Susan Dana-Jones. She is an expert in the area of biopharmaceutical development, vaccine product development, and also a lot in strategic planning. Welcome, Susan, to the show.
Thank you. Pleasure to be here.
Absolutely. I'm so delighted that we are talking today. And the topic for today's discussion is antibody development. Susan, you've been in antibody development for almost 3 decades now.
Yep.
And how do you see, how has the field evolved, and from the time that you started to today?
Well, when I started in antibody engineering, it was actually the early 90s, and that was a time when, antibody engineering was established but young. So monoclonal antibodies had been discovered, and people had quickly realized the potential of antibodies, monoclonals, to be therapeutics. And in fact, the first monoclonal antibody was on the market, OKT3, for transplant, to prevent organ rejection in transplant. But that was a murine antibody and had several issues with tolerance.
Yeah.
And so what was going on in the 90s was attempts to improve antibodies, make them more human-like, make them less immunogenic, finding different targets. And so really the field began to grow in the 90s, and products that we now know very well, Remicade, Rituxan, Herceptin, those types of products were all being developed in the 90s. If you contrast that to where we are now, we have engineered antibodies, antibody domains. We know what pretty much every amino acid in the antibody is there for, and we can adjust them as we need for the different therapeutic applications.
Absolutely, and that is where I think singularly more so has the therapeutic area played a role in how the antibody evolution has happened, or has process development been more a critical area?
Well, to some extent both, and in terms of the therapeutic area, what we've been able to do with antibodies as the field has grown and matured is, first of all, address more targets. We have better ways of discovering antibodies through different display technologies, immunization technologies, affinity maturation technologies, and then other areas in terms of the process side in antibodies is that as we approach targets where higher doses may be necessary for a more therapeutic benefit, we've had to establish very high production cell lines. The expression from CHO cells has gone from, milligrams per liter up to grams per liter in the last 20 or 30 years. And also in formulation, in ways to make high concentration antibody solutions for treatment of patients. So it's sort of gone hand in hand, the different therapeutic areas, pushing antibody engineering and process development into new technologies to enable treatment of new therapeutic areas.
And talking of new technologies, Susan, we are now having these platforms which are the CAR-T therapy, for example, or we have these dendritic cells or other platforms where we are engaging the T-cell based therapies. How do you see these growing or evolving? What are the challenges that we are facing today? What is the future that we are having as a perspective?
So that's a really, that could take hours to answer, but I won't take hours because I don't think we have that much time here. But so the CAR-T therapies have been very, very interesting, basically harnessing the immune system for treatment of cancers, harnessing the patient's own immune system. But they also are challenging in terms of the manufacturing. You're isolating the patient's own cells, having to transform them, put in the receptor, and then introduce them back into the patient. From the development side, that's a personalized medicine, that it's established, there are ways to do that. But other interventions, such as a T-cell engager or a checkpoint inhibitor, which is a single protein, a single molecule, it certainly fits more easily into the paradigm, the established paradigm, the bioreactors, the purification, the formulation, the administration across, all the patients that are suitable for the treatment. It's an easier path, and it's not an easy path. I'm not saying that.
It's definitely not an easy path, but there are two parts here, Susan. One would be, of course, in terms of scalability and patient response, and another would be regulatory response. How do you see both of these coming together in benefit of the patient?
Oh, okay. That's an interesting question. So again, the idea of using the antibodies to go in and activate the immune system and treat, especially in this case it would be oncology. It's I've seen some amazing successes. But at the same time, on the therapeutic side, you need to be very careful in just how much you activate and making sure that you're targeting essentially the right cells and not killing off other cells. There have been some headline grabbing incidents that were problematic. And so from the regulatory side, they're certainly aware of what happens in the industry, and they're looking out for patient safety, especially early on, and that's what they're going to focus on. From the process side, the challenges are different. The challenges for immunotherapies on the process side, they still lie in the areas of production, formulation, stability, the typical things that we need to think about in terms of making antibodies or engineered antibodies.
Absolutely, and I think all of us, we hope that we are going to make some quick successes, because there's a lot of patients who are waiting for all of us to make those.
We certainly hope so. I have family members who are suffering from different cancers, and certainly motivates me to go to work every day and think about how what I'm doing can affect not just my family members, but certainly can be helpful to patients all over the world.
Absolutely, I so agree, and I'm so delighted that you could come here and we could have this discussion. And I would love to have follow-on discussions with you on these topics and otherwise, and see how we can contribute to it, not only from the perspective that we go to work every day, but we can also engage with our audiences here and contribute to bringing what's going on in our side of the world as well. So thank you very much, all of you as well.
Thank you, Nubra, for having me today.